Hello, everyone. I apologize about that delay. We're good to get going now. So thank you for being here and joining us on the final webinar of our 2023 NECAN webinar series. The point of this webinar series is to highlight monitoring priorities in the Northeast, and this is our 12th webinar. And today's theme is new technology and rapid response. With the assistance of this series, and our presenters, the NECAN Steering Committee, will be working on the development of a regional monitoring plan for ocean acidification, and these webinars will serve as a resource for them as this plan begins to come together. Updates on this series are shared through our mailing list and on our website, so be sure to check out the website for all the past recordings and if anything gets added in the future. On the conclusion of presentations today, the Steering Committee will be asking this panel of presenters some questions, and then we'll open it up to a more general question and answer session. During that session, please feel free to use the raise hand function that is found on the bottom of the screen in the Zoom toolbar, or you can submit questions via chat, and our lovely moderator, Chris Hunt, will make sure that they are heard. First up, we're going to hear from Dr. Zhaohui Aleck Wang. Aleck is an associate scientist with tenure at the Department of Marine Chemistry and Geochemistry at Woods Hole Oceanographic Institution. He's a marine chemist with expertise in seawater carbonate chemistry, in situ sensor technologies, coastal carbon cycling, ocean acidification, and carbon fluxes in tidal wetlands. His current projects focus on studying ocean alkalinity enhancement as a CO2 removal, CDR, approach, the impacts of OA in coastal systems, blue carbon exports from tidal wetlands, and developing in situ sensors and sensor networks to study and monitor carbon cycling and OA. Aleck earned his Master of Science in Oceanography from the University of New Hampshire in 1998 and his PhD in Marine Sciences from University of Georgia in 2003. He is a scientist. He's been a scientist at WHOI since 2009. Please join me in welcoming Aleck. And Aleck, please feel free to share your screen and take it away.
Thank you, Austin. Good afternoon, everyone. That's a good, say a good few words about myself. I see many familiar names in the audience, which is good. So I'm going to share my screen now. Can you all see this?
We can see it, Aleck. It is not in presenter mode yet, though.
What about now?
That looks perfect. Thank you.
Okay, yeah. So I'm gonna, thanks again for inviting me here. It's always fun to talk to, give a presentation on NECAN. I involved in some early work with NECAN, and then been participating along the way. So today, gonna just give a little update on our sensor development projects, specifically focus on the high frequency and low cost in situ sensing of the carbonate parameters. The overview, I'm gonna give a little bit overview of the in situ sensing of the CO2 parameters. And then we're going to focus two technologies we've been developing. One is the channelized optical system, which you can make high frequency in situ measurement of DAC, PCO2, or pH at a time, one at a time, or we can actually make two measurements simultaneously together. The second technology we've been working on for the last basically 2 years is this low-cost pH sensor. It's targeting deployment on fishing vessels and gears, and then coastal studies. So related to the low-cost sensors, at the end I'm going to talk a little bit about this OCB steering workshop, and then I know there's people in this audience being into that workshop. It's happened early this year. The goal was to build a cost-effective coastal BGC network in collaboration with the commercial fishing community. The intent is to use some of the low-cost sensor available. So this is tied to the the sensor we've been developing over the years. So I don't need to give too much introduction on this slide. This is basically just the four major parameters, CO2 parameters, and with their definition. So we now have in situ sensors for all of them, but still, the commercial sensors are for the PCO2 and the pH. The DAC and TA may be close to be available commercially, but not yet, as far as I know. There's an emerging new technology or method to measure carbonate ion concentration, which is very useful for the calcium carbon saturation state determination, or so that's kind of direct measurement so that you can know the saturation state, and it's particularly useful for ocean acidification studies. But though I haven't heard about the in situ sensor yet, so maybe people in the audience already know that. So as a sensor developer, the first layer of question I'm asking is what kind of scales, what kind of the signals we try to capture. So on the left is a familiar schematic plots showing the spatial scales and the temporal scales for a lot of oceanic programs.
Chris and Dwight are here. Yeah, Adam too, but he doesn't know. Do a lot of questions.
Emily,
Sorry.
Emily, you're unmuted, Emily.
Oh, sorry guys.
That's okay. So yeah, so I highlighted this little square, which is basically you can see many familiar processes, and then what the carbon sensors and the carbon network sensor network can do at the moment. Mostly kind of covered in this square, so that's a range runs from maybe a minutes, hour to over a year, maybe tens, decade. That's getting a little bit difficult to detect, and then the spatial scale from meters to thousands kilometers, like in the BGC Argo program, that the pH starting to cover regional to basin scale phenomena. So on the right is model output. This the machine learning model give climatology seasonal signals for pH and omega and other CO2 parameters. So we can do reasonably good job nowadays to kind of project what the carbonate parameters look like in the shelf environment. But the challenge is still there's a lot of places that we don't have a coverage, so it's kind of hard to assess the uncertainty in the places where we don't have coverage, which is kind of the purpose of this slide. So we have for ocean observing, we have many challenges, the cost, robustness of technology, quality, the data, how you deliver data, and how you maintain the network and sensors and so on. So in the middle, you see this PCO2 sensor buoys around the United States, and the sensor obviously can cover quite a bit temporal variability at the site, but also we kind of lose this spatial coverage. That's kind of the gaps there. On the right, you see the kind of bottle sample coverage for DAC alkalinity over the last 3 decades. So some seasons, for example, summer, you have reasonably good coverage, but some other seasons, the coverage is not good. So you can argue that we have a lot of observational holes or gaps in the coverage area. So as a comparison, this is the weather station map that you can find online. That tells weather station coverage in the North America. So then and then obviously this is the order of magnitude higher coverage than what we have now. And then when successful the main successful story here is that they take this community-based approach that they spend quite a bit of effort and resources to develop low-cost sensors, weather stations. And so nowadays you can buy low-cost weather stations from different sources. They're shown in the picture. It's kind of mid-level weather station. You can buy it something on the order of $500. Then if you get a few $1,000s, you can get professional grade weather stations. So this is kind of the model I'm going to talk about. Talk about later that using fishery-based opportunities to maybe use that as opportunity to make measurement of carbon parameters. So that's the kind of the spatial coverage challenge, and then for CO2 parameters, there's a couple of very specific challenges. So the first one, obviously, we know the signal dynamic range is so large, so the OA signal is small, tiny. The pH is only 0.01, 0.03 per year in open ocean. But natural variability is large, cover a wide range of temporal and spatial scales. And then everyone knows that we also need at least two CO2 parameters, and which parameter pair is important. The pH and PCO2 or FCO2, commonly measured to give estimate of CO2, other parameters of CO2 parameters, kind of large uncertainties. It's not desirable in many places, in locations. And then, so all of the method we've been having for in situ sensors, in carbon sensors, the response time is relatively slow. So this is limit some capability we can deploy on a mobile platform or in a very dynamic environment like coastal systems. And then many of the sensors require consumables like reagents, so that moving parts which make the sensor relatively complex. So those are some of the challenges with facing. Though the last few last few decades, I've been working on the in situ sensors, and this will give you a kind of update what we are, where we are at the sensor carbon sensor field. So you can see there's variety of different methods being used to make in situ measurement of the carbon parameters. Okay. Then there's a trend that over the last maybe 5 years we start to have solid state sensors. Okay, so there's from PCO to optode, and everyone's familiar with the ESFET, CFET, pH, and also now we have pH optode that emerging. So there's also the ESFET pH-based alkalinity sensor nowadays, and this is on the researcher stage. So we are being working on a new solid state DIC sensor, but this is kind of the a very early stage of that. So another good development is that for all of the parameters, some of the sensors can reach really or near climate quality measurements, which is very nice. Sensors can be deployed in a variety of different platforms. So it depends on the characteristic of sensors. So that's also very encouraging. So for the low-cost sensors, there's the community has been working on this for some time as well. This is a review paper we published back in 2019. So kind of give a review of the low-cost sensor in particular about geochemical sensors and biological sensors. Okay, so here on left a few examples. There's a DO sensor, relatively low cost, and then nowadays it's commercially available. Then there's a pH sensor in situ as well as the handheld. So one of the conclusions from the paper is that particularly for DO and pH sensors, we feel it is relatively mature to be able to deploy in a larger scale. So we've been working on kind of try to make that a reality in the recent years. So here's the channels technology. We've been developing this technology for some years, and this is already a second version. Okay, so the development goal is so that we can measure two parameters simultaneously. In this case, we particularly target the pH DAC or PCO2 DAC pair. Okay. So we try to do it in high frequency, and so that this can be used on mobile platforms, profiling purposes, and time series. So all of the parameters are based on the spectrophotometric detection, so that the module can be swapped between different parameters, so make it easy to adapt for simultaneous measurements or individual measurement. And the sensor has the in situ calibration capability, so that we can check the quality of measurements during deployments. Then the current sensor can be deployed up to 2,000 meters, 1,500 or 2,000 meters. But really, there's no limitation that the measurement can be in full water column, so it can be deeper. So this is the pH spectrophotometric pH principle, and many of you are familiar with this. Basically, we have the pH sensitivity indicator added to seawater, and then the dissociation of the indicator have very distinct color for the base and the acid form of the indicator, and then the base, the ratio of the base and the acid peak in the absorbance is related to the sample pH and along with a few other parameters, constants. So the method can be calibration free, which means that during the field deployment, you only need to measure the absorbance of the indicator, and then assuming the indicator doesn't change, it's an optical property, and then the dissociation constant is doesn't change, then you don't need to recalibrate it in the field. That's but we still think it's important to check during the deployment whether there is some changes in terms of optical property and property of the indicator. But by theory, it should be calibrate free. The DAC method is somewhat similar, not exactly. So in this method, we first acidify seawater samples and then convert all of the carbonate species to dissolved CO2. And then we let it flow along with, outside this CO2 equilibrator. In this case, we use the Teflon tube, which is permeable to CO2. And then inside the Teflon tubing is this the pH sensitivity indicator. And then we direct the sample flow the opposite or counter flow with the indicator flow, which increase to make the response time faster. Okay. Then so the CO2 will equilibrate between the acidified sample and the indicator, and that will change the color of the indicator, basically the pH of the indicator. As after equilibration, we can direct the indicator flow through the optical cells for the pH detection. And then you can see the equation that basically the pH of the indicator is related to the equilibrated CO2 fugacity on both sides, and then that can be further related to the DNC concentration in the sample. Okay, so that's by theory again. This if the indicator does not change, And then this measurement should be cavitation free. But again, we still check that. But we have more and more confidence during the field experiment that this actually holds. The cavitation free is something that can happen. So this is the first generation of the channels. So we have a couple of papers published. So the measurements, we do like this every 15 minutes cycle. And then, within that 15-minute cycle, you can achieve 8 minutes continuous measurement. And then the sensor was built for surface deployment. The one major hurdle for continuous measurement for this version was, we used the syringe pump, so which very precise. However, you do have to refill your syringe every once in a while. So that's why we have this kind of 15 minutes measurement cycle, the 8 minutes for continuous. So the measurement quality, at least the screen, and you can see is very close to the climate quality during the field deployment. So for the second version, so we just did a couple of years ago, we did some tests. This is a time series test in the Cassett River. It's one of the tidal river on Cape Cod. So the total deployment days about 59, so about 60 days deployment. Then we test those continuous mode and the intermediate mode. So here is the data, so the kind of full data. Sorry about the small size. The blue lines are the DAC measurement. The red circle is the bottle samples. The black is the salinity, and the red lines are the DO. So you can see immediately see that the covariability between the DAC and the salinity, which is basically a tidal driven the signal, right? So and then there's this kind of opposite variability between DO and DAC concentration, right? So that's clear. So this all makes sense. Some of the I highlighted two events during the deployment period. One is the red square, which is basically a tropical storm passing through the area, and then the other is the blue line, which is basically the cover. There's a northeastern storm passing through. So the bottle sensor comparison, which is at least the right left corner, and it's reasonably good. So it's not as good as the first sensor, first channels yet. There's some more further improvement on the optical measurement. We think we can get that improved further. And here's the kind of interesting highlights of what a sensor can do during episodic events. Two storms. The two storms. All right, Austin. Yeah. So I need to really go quick. So the two storms. One is the tropical storm Henry, which is give you kind of this change of the DAC and the TO. It's a major. It's a wind storm, so it doesn't have a rain. So in that case, the sediment being stirred. And then you have CO2 DAC and high DAC coming out from the sediment, and then there's DO has a low DO concentration, and then the northeastern is primarily a dilution effect, where you have a lot of rains and that dilute the DAC, but doesn't change the DO content much. So I can move quickly on this. We also did this surface towing experiment or deployment in WHOI Bay. And then here's the data showing very high resolution surface map, and we also did some deployment for the shelf, and then that's try to look at the deep core environment using channels. This is showing some high resolution DAC profiles. So quickly, the low cost pH sensor. So we tried to develop this low cost for high frequency measurement and targeting for fishing vessels and other similar platforms. And then we try to integrate this pH sensor along with the low cost DO and CDD so that we can deploy on fishing vessels. And then the whole package will have wireless capability so that we can relay the measurement near real time to land. So that's the idea. So and then it is still the pH is spectrometric measurement. And then in this case we try to do a little bit differently. So the whole package is small and relatively simple. And then we'll try to achieve the low maintenance and capable to doing high frequency profiling. And then the depth range up to 300 meters. The cost is on the order of a few $1,000s, and right now it's kind of 2,500. So that's the goal. And then we did the bench testing, which is shown here. I mean, you can see the size of this. It's relatively small. The red box has the basically sensor components I showed in previous slides. So it's relatively small with two little pumps on the table. So the plot showing the residual of repeated measurement from the mean. So you can see the repeatability is pretty good. On the order of 0.002 pH unit. So then they will still need the lab calibration, but there's no requirement for C2 calibration. That's at least this is the design. And the sensor has self-referencing for the optical signals, so they count for any drift. So then there's no need to correct the drifting during deployment. So in recent we just started a new project with NECAN Vandemark. Try to deploy the sensor on fishing vessels. In this case, in collaboration with the EMOTE program. So we've been doing the seawater temperature in Northeast. You can see the coverage pretty intensive. And I feel the fishing gears we're talking about is here. You can see that we're thinking about put the sensor on the lobster traps at the starting point. So that's being planned for later this year and into the next year. So we're very excited to see this. So a little bit just this is a working steering workshop. Okay. So the goal is, as I mentioned, is to bring everyone together. Try to make a plan and then to establish a fishery-based BGC observing network in the Northeast. There's 60 people plus some folks from online, and we'll try to leverage with the decadal long experience from the infrastructure, deployment of the physical sensors in the coastal water, and then we try to add the new BGC sensors, low cost BGC sensors into this network. So quickly, this is the last slide. So this is the kind of the existing program in the Northeast. This is the primarily measuring seawater physical properties like temperature, salinity. And the longest program obviously is the EMOTE. On the right, you can see this almost over 3 decades. You can see that this EMOTE program captured the warming of the seawater in the region, which is very promising. This is a very successful story, both at the surface and the bottom. Okay, so we're gonna try to use put our sensors into this network, and then hopefully this will start a community-based observing network in the Northeast. I can skip this. We talk about a lot in the workshop. So if you're interested, please go to the OCB website, and there's a workshop link there. So at last, we'll just leave this summary here. So the one last thing I want to mention is availability. That's required by you guys. So at the moment, the channels can be built for individuals through WHOI contract. So we're in the middle of seeking commercialization as well. So currently we are collaborating. We are building one sensor for channels for Northeast Fisheries Science Center. So they'll try to deploy it on the HAP, CAM, and ECOMON cruises. So this kind of direct link to in situ measurement, in situ chemistry measurement with the in situ biology, you know, especially the bivalves. And then the other is you know, we just mentioned we are collaborating with NERCUS and NROC, try to deploy the new pH sensor lobster traps. Okay, with that, thank you for your attention, and I'll just leave it here, and if you have questions, we can come back. Thank you.
Thanks, Aleck. So what we're going to do, as I mentioned at the top, we're going to circle back and we do one large question period at the end. So we're going to move right along to our next speaker. So our next speaker is Dr. Doug Vandemark. And Doug's research interests lie in air-sea interactions with a focus on satellite ocean remote sensing of wind waves and air-sea exchange of momentum, heat, and mass, including greenhouse gases. His specific research in these fields involves microwave oceanography, including altimetry, scatterometry, radiometry, and synthetic aperture radar, as well as in situ measurement techniques and instrumentation related to turbulent flux parameterization, gas transfer, and wind wave interaction. His recent research focus is on salinity, remote sensing, as well as investigation of carbon dioxide air-sea flux and its remote sensing using satellite ocean surface topography, wind, temperature, and ocean color measurements. This work includes developing and operating observation systems, platforms, and sensors that support monitoring in coastal and offshore waters. Please take it away, Doug.
Okay. Trying to share here. All right, there we go. Okay, does that look all right?
That looks excellent.
Okay, so my understanding of the marching orders for today was just to give you kind of an informal update on some of the things that we were involved in this year and are still involved in terms of what I call here the Tripos event. And do I have that right, Austin?
Yes, you do.
Okay, and I have maybe like, you want me to keep this to like 15 minutes or 10?
15 minutes would be wonderful. That would keep us excellently on time.
Okay, and I see we have a couple other speakers that look like will be on a similar topic, and so maybe I'll help set them up a little bit, and I've got some things in here that maybe I can skip through based on the other speakers that are coming. And feel free to break in if you wish, but I guess questions reserved to the end is what I'm hearing. So as I said, this is just an informal grab of some slides related to this event. And essentially, as maybe some of you know, and one thing I'm assuming with this group is everybody's familiar with the Gulf of Maine. I don't really think I have very many maps here in the beginning or anything. So if somebody gets lost, please do just speak up. But at least for what I would try to focus on today is basically what got our attention this May, April was the first two items here. We saw the carbon dioxide at the buoy that we operate off of the Isle of Shoals, about 10 miles offshore here in New Hampshire, start to drop to very low levels during the spring bloom. And that's, you know, that's always interesting to see, because some years in the last few years, we've had fairly weak blooms. And to see a good one was, you know, an encouraging sign maybe for the zooplankton and the upper trophic level feeders. And but then it really started to drop out to go to very low levels we hadn't observed since we've been doing this really since 2004. And at the same time, there was a bunch of brown in the filters, brown gook. And it turned out that this was essentially what we usually know as serratium. And both of those things were really quite unusual. And so that got our attention. And as up until say mid-May or something, the blooms usually drop out. And so we thought, oh, this is really kind of an unusual and really strong bloom. Let's get on it. And the third thing happened over the course of the spring into summer, that it didn't stop. And so those are the three unusual things that I tried to talk a little bit about today. So from the carbon dioxide side, this is just a kind of a screen grab of the time series of oxygen. Let me get rid of that. Over the course of the accepted deployments at PMEL, we have a gap here that I guess I didn't know they'd put in there. But you can see that basically this is what got our attention. This is this spring. And this is my very messy spaghetti plot down on the bottom that I made of all the years, roughly from, I think this is the end of February through to August, of all the years of measurements. And this is, I think, a smooth, like 1 to 2 day smoother on the, this is the difference between the atmosphere and the ocean PCO2 near the surface. The black line is the average of all the years in our buoy data set. And this red line, when it dropped here, I got excited because, wow, that's well below anything we ever saw. And then it kept going, and we were very interested right in here and going, geez, this has really been going on a long time. This is really interesting. And so by the time it got to here, it started to become kind of unsettling. And so this is the kind of the carbon side of the story that got our attention, and it has led to some of the things that we've been trying to do here. Let's see. So at least in the Western Gulf of Maine, I just did some back of the envelope things this spring and then into the summer, that the sink of carbon dioxide from the atmosphere into the water is at least 2 to 3 times anything that we've observed in the past. Just in terms of DIC associated with these organisms and the fact that they kept reproducing and kept shooting organic matter down into the deeper layers, we're estimating that it's quite a lot of excess carbon. There's still a lot to look at with our bottle data. We haven't run it all, and so it's very preliminary to say more. And then in terms of the carbon overall, obviously the fate of this carbon and how big of a footprint this event made is still to be determined. And one possibility is all just going to blow back out in the fall. And so that's one of the observational focus that we're trying to maintain going into the fall. So this was at our buoy 10 miles offshore, which is, right in this area here. We also do a monthly cruise out to Wilkinson Basin that's about 40 miles offshore in this area. But we were interested to know, how broad spread this thing was. And so I'll talk a little bit more about our communications up and down the coast in a bit, but at least we knew that are familiar with the NOAA Ecoman surveys, and they performed one in late June and then again in late August. And this is the June data here. And as I note up here, when we were out on our ship in June with our flow-through system out in Wilkinson Basin, we saw a level of 110 microatmospheres, which is, we've never seen a level lower. And so we were taking pictures of it and thinking, oh, this is really cool. And we thought, we've got a record here. And then like less than a week later, Ecoman went out, and I'm pretty sure, I haven't seen their raw data yet and their process data, but I think they beat us. But the bottom line is this dark blue here, all of this is the whole Gulf of Maine inside of the north flank of Georges Bank up to the mouth of the Bay of Fundy, around basically the 100-meter isobath was just carbon dioxide depleted in terms of near surface atmospheric CO2. And this gave us an indication that since we haven't seen these kind of levels before associated with a normal diatom spring bloom, for example, that this is probably the same organism, the same species, the same process drawing it down. And then this is the August data, just to button up the Ecomon. This was their survey data. And you can see even on August 24th, they're still down at 300 microatmospheres out in the middle. And there's still green water out there in the satellite data. So just to back up this Gulf-wide part of the story, this was middle of July, which was really the first time that the clouds cleared. I don't know if you remember back to our lovely June this year when we never saw the sun. But for most of May and June, we couldn't see the Gulf of Maine from space. So we thought there was probably a very strong phytoplankton bloom going on out in the middle. We really couldn't see it from the satellites until around the middle of July. And these are a couple of images that Kim Hyde and colleagues pulled together, showing that this is the anomaly over here, the amount of excess chlorophyll with respect to their average for this period in July. And this kind of shows this western half of the Gulf of Maine focus associated with this particular organism. They built an algorithm specifically to try to trip on this particular dinoflagellate species, and this is what the mapping of it was on the 23rd of July. So it wasn't just in the western Gulf of Maine, it wasn't for a couple of weeks, it seemed to be broad spread, and it seemed to last a long time. And just another quick data poll that we were watching during this period. This isn't the full map, but this is Mark Baumgartner's glider out of Woods Hole for right whale listening. And I was very interested to see these data through April through early August. This right here is a map of chlorophyll. And so I found this interesting to watch during the period. You can see a very strong surface chlorophyll bloom, but you can also see chlorophyll down to around 70 to 80 meters out in this Wilkinson Basin. This is Georges Basin into Wilkinson during this whole time series. And so this is another indication that it didn't terminate, and it basically looks like the same bloom data. And so just to go from the carbon to what was doing this, we were calling this serratium for the first couple months that we were doing this, because this is what everyone in the Gulf of Maine knows is serratium. And then Greg Teagarden that works with Jeff Runge, we were talking with them and asking them if they'd seen it. And he came back and explained to us that this is the species, this is Tripos malari. It was renamed like 10 years ago or something based on DNA. But this is what was doing the the carbon drawdown. And I think that a couple of the speakers after me may talk more about this, but typical levels we've seen are of the order of 1,000 to 2,000 cells per liter is a pretty high sampling of this organism. It's always around in the Gulf of Maine, pretty common at least. But to see it at 2,000 cells per liter, it will be the king in the summer in the subsurface chlorophyll max, and that's quite a high level. But we were getting levels that were quite significantly higher than that. This is another of these filter draws, like the first picture I had on the first slide of a net tow. This is Sean Shellatow holding some of this stuff on a cruise we were on the 17th of June out in Wilkinson Basin. And this is FlowCam image of the same organism from Mike Brosnahan's Radbot. This is one of his first images when he first got out at the Isle of Shoals, and the first thing he saw, and the only thing he saw pretty much for the whole time it was out there, were these guys. So they were pretty much dominating the scene from mid-April to at least late July. And to get back to the cell counts, this is the record of some of our visits to the CO2 buoy site that I mentioned earlier. And you can see we were getting numbers up to 60,000 cells per liter. This red line is a mark near 2,000 cells per liter. So you can see all the way down to the depth of that buoy, it's around 70 meters of water. We were seeing levels that exceeded the usual level of this organism, maximum level, all the way down. And it stayed this way through this period. And the near to the coast, because of the DES and other HAB program monitoring work along the shore, there's quite a few time series of this near shore. And in general, the near shore bloom was really localized more from April into May, and then it tended to drop down from what we've seen. Very near shore. I think MWRA will have a different story to tell. So overall, with this bloom, number one, it was so long lived that we don't know that we want to call it a bloom, but we have a lot of things that we could and people are interested to discuss, including the initiation, the maintenance of this, how it survived for this long a period with the nutrients available to it and the normal thought of a phytoplankton bloom and decline. Why did it end? And then what is the fate of all this this carbon, and how broad spread was it? And so I think this is some of the rapid response discussion we may get to have today, so I don't want to take up more floor time on this. I will say that late in April, this was significant enough that we tried to get an NSF rapid funding proposal going, and that did not get funded by NSF. And maybe one of the discussions we'll have is everyone has limited funding in their normal portfolio of what they can sample. And so once that failed, we didn't really have a lot of ideas on where to turn. And so we just basically tried to ramp up our own internal sampling at the buoy to at least capture the event as it kept unfolding. And I believe that just in terms of possible fall work, I'm not exactly sure of the status of it, but I know Kai Ziervogel was working with Syracuse on the idea of getting up with UConn and looking at the possible deposition of the dead tripos on the bottom, and whether they got there or whether they were chewed up in the mid-water column. So in terms of a big implication we were concerned with at the end of July going into August, it was oxygen. In terms of if this material was that abundant and decomposing, were we going to see anything like the only other large bloom that we know of in the literature? And I've called up and down the coast a little bit, and this is the only thing that has come up so far on the East Coast, is the 1976 New York Bight bloom that actually went anoxic in the New York Bight and took, I think scallops were the big loser in that event. And I think that, as I say, I think MWRA probably has the best data set to talk about this, and I think we'll probably hear about that today. So I don't want to steal their thunder, so I'll sneak through there. But the Ecomon crews did go out in both June and August, as I mentioned, and Chris Melrose sent us up the CTD cast last week from the August cruise, because we didn't know how much concern we should have in monitoring oxygen going into the fall. And this was kind of the worst case cast here. This is depth on the y-axis, and on the x-axis, this is oxygen saturation. And oxygen data are always preliminary, but these look pretty, it looks pretty well calibrated. You can see near 100% up near the surface, which is what it should be. And the lowest levels they were seeing are down near 60% at 150 meter depth. And I think the lowest that we're seeing in our cast, again, preliminary, is around 55%. And so overall, this cruise over the whole Gulf of Maine, and I looked at all their casts through the Gulf, this was the lowest. And so I feel that the emergency on oxygen may not yet be there. It doesn't mean that it couldn't be coming, but at least as of late August, things don't look alarming. And so I just have one last slide just as ideas for discussion later. I think the whole event has brought up some things about community ocean observing and group communications. And, you know, one thing that came to mind for me is this wasn't really a HAB. And so at that point, what is the response on an event that we don't necessarily have an organization that's really set up to go after? And at least this event may suggest that we do or don't have such an organization. But and then obviously a couple of these other questions I have on the slide you can read for yourself. So that's my little observational notes, and I hope I kept within the time or thereabouts.
Thank you, Doug. Next up, we will hear from Dave Wu. Dave is a senior program manager at the Massachusetts Water Resource Authority, or the MWRA, a regional public water and wastewater utility serving the metropolitan Boston area. He has been in charge of the MWRA's environmental monitoring efforts in the Massachusetts Bay, Boston Harbor, and the harbor's tributary rivers since 2021. Previously, at MWRA, he has worked on permit compliance, impacts of combined sewer overflows on water quality, and fish and shellfish biomonitoring. Dave has a bachelor's and master's degrees from Duke University. Please share your screen whenever you're ready, Dave.
Thanks, Austin. Let's see if I can get this right. Okay, let's see. Slideshow. Okay, can everybody see this?
I can see it perfectly. Thanks, Dave.
Awesome. All right, so this is a slightly different kind of presentation from the two previous. Thanks both you, Doug and Aleck. This is more about sort of the, I don't know, it's not science, and I don't know if I call it admin either, but it's kind of about our experiences with rapid response monitoring. So to have response monitoring, I feel like you also need to have a routine monitoring program. So I've tried my best to cram our routine monitoring into one slide. This doesn't necessarily capture all the details, but I think it does get the basics. So in Massachusetts and Cape Cod Bays, and we consider those two water bodies separately, we collect sort of a standard suite of physical parameters, DO, you know, stuff like that, nutrients, phytoplankton, zooplankton, Alexandrium and Mass Bay only. And so this is 9 times a year, and this is done by contractors. Right now, those contractors are Battelle in Mass Bay and the Center for Coastal Studies for Cape Cod Bay. So that monitoring program is actually prescribed in our NIPD's discharge permit for Deer Island, the Metro Boston and our sewage treatment plant. We also do monthly bacteria monitoring for fecal coliform and enterococcus out in Mass Bay. That's an in-house program. And the ones I'm quite familiar with are our Boston Harbor and Rivers programs, which are also done in-house. And we're collecting standard physical parameters with SONS, water quality SONS, as well as nutrients and bacteria. For out in the harbor, it's 2 times a month. And in the rivers, it's numerous times a year. We probably have about 60 trips out, at least, out to those 3 river basins. And we also have a rivers nutrient monitoring program that's twice a month. So our first and probably most expensive rapid response program is for Alexandria. So these are sort of triggered 2 different ways. Alexandrium samples are collected at the green square stations. Those are routine water column stations 9 times a year. So when there's a cell count threshold met, that's if the counts are over 100 cells per liter, that would trigger a rapid response survey. And also the other trigger is PSB toxicity levels. And we get that data from Mass DMF. So once a rapid response survey for Alexandria is triggered, these are weekly surveys that go out and they will sample at all the stations located on that map. So the routine station is routine stations in the green squares, as well as a number of sort of light green circles that are sort of the Alexandrium stations. So this is all done by our contractor, currently Battelle. The data collected includes cell counts for Alexandrium, nutrients, as well as some pigment data, chlorophyll and phaeophytin. This can be a very challenging program. This is something that's not done in-house. So there's some sort of contract and administrative overhead that we have to do. This is all done under task order in our contract. The timeline is very compressed. It can be just a couple days. It's usually just a couple days from when we know the results from the routine survey to when we need to set up a weekly response survey the next week. And it always happens over the weekend. I've learned in working for a public utility that nothing happens on the weekdays during the workday. It's always on a weekend, and it's always after hours. And as you can guess, it's also unpredictable. For example, this year we had no Alexandria rapid response surveys, and that has not been the case for the past few years. Last year we had 3, and I believe the previous year we also had 3. And then to change gears, this is more freshwater, but I think it's still safe to talk about this. From 2016 to 2019, we had this sort of, we nicknamed it the storm chasing program. And after a rainstorm, we would do 5 days of intensive sampling in either the Charles or the Mystic River basins. And this was to evaluate effects of combined sewer overflows if they occurred, and stormwater discharges on bacterial water quality. So the focus here was on bacteria. In this case, Enterococcus and E. coli, which are the 2 indicator bacteria for fresh water. So this was part of our CSO assessment, which is a sort of a 20-year CSO control program. We're trying to reduce CSOs in the Boston Harbor and its tributary rivers. And I realize that this is not the audience I'm used to talking to about this. So I guess I should say that if you don't know, CSOs are sort of a legacy of 19th century sewage system design. There's the stormwater and sanitary sewers were combined together, not separated like they are now. So usually when it doesn't rain on a day like today, there's no problems. Everything just flows to the plant. When you do get a lot of rainfall, you will overwhelm the system. And usually to prevent any backups at the plant, you'll send out both sewage and stormwater out and outfall into the rivers. So generally the sewage percentage is pretty low. We think it's via our collection system model, we think the sewage percentage of these overflows is generally less than 2%. But there's still some concern about bacteria, obviously. And so it turns out the data that we used in this program was used to calibrate water quality models for the Charles and the Mystic Rivers, done by our consultant AECOM. So this program had a number of challenges. The good thing it was done in-house, and staff time is free. So we didn't really have a whole lot of issues necessarily with sort of getting the paperwork done and everything like that we did with the task orders for Alexandrium. But this is highly weather dependent. Weather prediction obviously is an art as much as science. So my project manager and I would spend a lot of time poring over weather forecasts, talking to people who know a bit more about weather than us. Is the rain actually going to come, and is it big enough to make it worth our time to mobilize a bunch of staff to do this sampling? And we also did it over weekends as well. So staffing could be an issue. Sometimes we have all sorts, and this goes in with the next thing. The lab has a number of other existing projects. So we had to balance, can we put off these things? Can we put off these other projects to do this? And if we do it, do we have enough staff to cover one or the other? So that was an ongoing challenge. And unfortunately, we have ended this program. It provided us a lot of very interesting data, but we just didn't have the manpower and the time to continue it after 2019. And our other in-house program, and this is kind of back to the Bay. is bacteria monitoring for Massachusetts Bay. When Deer Island was built, part of the Boston Harbor project was to build this 9.5-mile outfall, which you can sort of see as the dotted line and the red line in the middle of the graph. So this treated wastewater was discharged out to the bay rather than to Boston Harbor, and so there's some concerns from the DMF about bacteria and its potential impacts on shellfishing. So we have an MOU with DMF to do monthly monitoring for fecal coliform. We also collect enterococcus at the same time. And this is a regular monthly thing, but we also have a rapid response survey that is triggered under special events that we call the adverse conditions survey. So one of the conditions is disinfection failure at the plant. So at the plant, there's a chlorination and dechlorination system. And I want to stress that this has never happened. This is just a contingency. We don't expect it to happen, but you never know. So the main trigger for this is extended blending events from large rainstorms. To keep it simple, when there's at very high flows at the plant during storms, we will partially bypass secondary treatment and combine sort of just primarily treated effluent with secondary treated effluent. That is disinfected, and that's considered a blended effluent. So that's discharged when there's a lot of rain. So when that happens, there's an additional survey as soon as possible. And that's been going on since 2000. And we probably get these adverse conditions surveys, maybe one every other year or so. I doubt it averages out to one a year. But they're not rare, but they're not common either. So the challenges for this one, as you can imagine, since these things are generally triggered by large storms, hurricanes, tropical storms, tropical depressions, and the like, is weather, because safety is paramount. We're using a small boat to do this sampling, and that can be a real issue. We do tend to get out within 3 or 4 days, but immediately it rarely happens just because of the safety issues. Staffing can also be an issue depending on the timing. This can happen anytime, weekends. Maybe their staff are on other projects. And there's also a boat availability problem. We do have a contract boat available that can handle slightly rougher seas than our in-house boat, but that boat also has to be available as well. So I'm going to wrap up with this slide. So this has a bunch of links, and hopefully this PowerPoint will be posted somewhere. This has a bunch of links. One is our quality assurance project plan for routine monitoring, which people may find interesting. The plan for rapid response surveys for Alexandrium, sort of the water quality model results for the Charles and Mystic Rivers that were developed from the storm chasing data that I mentioned. And there's also a report which sadly is slightly outdated, just on the DMF bacteria data. And what I did not put on here, because I was kind of in a hurry to put this together, is my contact information, which I will put in the chat. But Doug mentioned sort of the MWRA data set. We're obviously a public utility, so any information we have is public, and we are more than happy to share it with everyone once a formal data request is submitted, and that can be emailed to me. But again, I'll put my email in the chat, and during the discussion, we can perhaps talk about what we have available and kind of how fast we can turn it around. One thing I wanted to say in response to Doug is that our data is not necessarily rapidly available. For example, the phyto and zooplankton data I think we're basically up to about May at this point, loading it into our database. So this is not, we don't turn these things around immediately. We go through a lot of QA/QC before we consider it final. So on that, I think I'm done, Austin, and I'll turn it over to you.
Thanks, Dave. That was a wonderful presentation. Moving along, unfortunately, Cameron Thompson had to jump out to a childcare conflict, but luckily Jake volunteered to convey some of the larger takeaways of what he was hoping to convey. So Jake, jump in.
Yeah, thanks, Austin. I don't have a lot to say. Like Austin said, and the NERA crew staff, we're all kind of on notice that Cameron often has afternoon childcare responsibilities that can change on a dime, so he had to step away. He did note that he expected that Doug would cover much of what he had to say, and I think he did. He left me with 2 points to pass along. One was just to make a note that at the moment, our crew at Syracuse and the team over at UNH are working together to plan a fall and maybe spring seminar and workshop program. And we see this as being kind of part of this rapid response. So the rapid response is what goes on in the water and in the labs to, to monitor, to collect data, to start to analyze it, but also sort of how we organize communities of science, how we ensure shared understanding of what we're learning, and how we start to develop some kind of an emerging consensus. So we don't know exactly what that program's going to look like. We're trying to figure out the when and the what, but Cameron thought it was worth noting that that sort of off the water activity is part of the TRIPOS response as well. The other thing he noted that I thought was a good point was that the work that we've been doing at Syracuse, at least, to kind of contribute to this TRIPOS rapid response has largely been sort of under, we're looking at it under the banner of the Integrated Sentinel Monitoring Network. We've had some funding set aside for new analyses for ISMN that we're kind of diverting to support this evolving rapid response. But I hadn't thought about the significance of that. I was glad Cameron pointed out, because we at times, I don't think Steve Kucher's on, but Steve is often one who's sort of reminding us that there is overlap between what NECAN does and what ISMN does, and including many of the people involved in each. So I'm not sure what that means exactly, but I thought it was just worth noting that the TRIPOS response on our end is kind of happening under the banner of, to some extent, and using resources earmarked for ISMN, but the ISMN NECAN kind of overlap in interactions or something we talk about periodically, maybe not enough, but maybe something we need to talk about more as we, through the monitoring plan, start to think about more systematic rapid response strategies. I'm just scanning my quick chat with Cameron. Those are the 2 main points he said. He said he expected Doug would cover the science pretty well, and I'm inclined to agree. So that's it.
Thanks, Jake. And now I'm going to pass the floor over to Chris Hunt to moderate our question and answer session.
All right, thanks, Austin. Thank you to all the speakers. They're really great. Well, obviously we have 2 kind of different topics, so we want to make sure that we get to questions that address all the speakers and what they presented. And the steering committee members have been keeping sort of a running list of questions, and I'll refer to that, but I totally hope the steering committee members will come on and ask their own questions. But I'm going to start off, because I have the floor right now. I'm going to start off by circling back to Aleck. And I sort of wanted to think more broadly about your channel sensor and also the pH sensor that you're developing. But I'd like to get your thoughts about what are sort of some of the challenges in commercializing and making a new technology broadly available. And I'm thinking in particular about the CFET. Those of us who are CFET users have had a sort of rough go of things the past few years. And so what do you see if you wanted to bring something like the Chanos or your pH sensor to wide adoption? What are some of the hurdles to that, and what are some of the things that need to happen?
Yeah, thanks, Chris. That's excellent question. I don't have a full answer to it. I think it is difficult, but I can throw in a couple of things or options I'm thinking and actually starting to do. One option, I mean, everyone knows we have a relatively small market.
Yeah.
So at any given moment, commercial company comes in, they look at this market. Yeah, gee, we're only going to sell 10 of them this year or 20 of them next year. That's not gonna make too much of sound. But I think the community is moving towards this so-called open source. kind of mine site. So we have, my students actually, working on this low cost pH sensor. He has this low cost pH sensor, which is still kind of under the IP of WHOI. But he also started another one kind of similar, but it's open source. So he collected some get some donations from outside source, and then start to build this handheld pH sensor. And in principle, it's very similar to what we built in the in this IP protected WHOI IP protected world. And it's a starting point. I don't know whether that's going to be the significant things coming up, but I do see a lot of people very much interested in this kind of model. So which really means that anyone look the open source, they could build themselves. Some people may use this as a opportunity to make money, but I don't know. I mean, that's another discussion. So that's one point. So I want to make. Oh, do you have another question for?
Well, can I follow up a little bit? So
Sure.
I really like the idea of open sourcing, something where like maybe the pH sensor uses a bunch of, off-the-shelf components or whatever, or it requires, only a limited amount of fabrication or something. But something like the Chanos seems like that's a pretty big undertaking for somebody to build themselves, right? And it seems like maybe you're taking a little bit of a different approach where you're building through and providing them through WHOI and using WHOI as kind of a vendor. Do you think is that the path that you're pursuing for now?
For limited number of units, I think that is a way to do it. So obviously WHOI has overhead, which is kind of high. Yeah. But on the other hand, it doesn't have the profit. We don't really charge. I mean, we just charge the labor, whoever build this. And then once it's a nominal cost. So I did a little calculation. If it's commercialized, this usually what's the profit margin versus if I build this at WHOI. So WHOI is okay. Let's say for limited number of units, including channels, and probably we're definitely going to include the low cost sensor in the future, which is basically just budget for fabrication and then people's time. And then the design is already there, so we just kind of don't charge any engineer design because this is already been developed. So in that model, so for a small number of units, that's probably okay. But there's always the upper limit. I mean, how many units WHOI wants to do this without commercialization? So
great. I know Dwight wrote a couple of questions down too. Dwight, I don't know if you want to hop on or somebody else does.
Yeah, sure. Can you hear me?
Yeah.
Yeah. So Aleck, I'm actually really glad you brought up the open source aspect, because that's something we've been thinking a lot more about. So because we've been running into, as a program, we've been running into real challenges where we're not getting the support we need from the commercial vendors. And we're finding that BGC is always this boutique community. What we do is we spend often millions of dollars in developing a technology, transitioning it to the commercial field. The commercial field makes a profit until they saturate the market, which happens usually pretty quickly because we're a small boutique community. And then they immediately bail on it, and we have to spend millions more to retransition it back into the agency. Yeah. So I'm really wondering if we maybe think we need to rethink how we do this. And one of them is open source and distributed manufacturing. Like maybe we need to be bolstering the 3D printing capabilities of some of the universities around the nation to try to ensure that. And then when you're developing a technology, you're developing it with open source and distributed manufacturing in mind. Like, not how am I going to build this for commercial production, but how am I going to build this specifically so that the community at large can build it at cost. So anyway, so that's one thought, more of a comment, and maybe that gets to Chanos and how could that be made in a manner that made it suitable for open source or distributed manufacturing? Maybe I'll ask that. Any thoughts on that? Or has it already been being built along the path of envisioning some company's going to pick it up?
Yeah, for channels, because I mean it has built-in IP. So I don't know whether what WHOI would think if you just do open source. But that's a conversation. Maybe there's a parts that can be open source, which is commercially available already on the market. And then what really is that someplace you put components together.
Right.
Has the sensor built. So in that case, maybe that will lower the overall cost of the building the channels. So that's one thing. And the other is that we actually try to improve the something like channels using readily quick 3D printing and prototyping processes. So some of the early more expensive components already been replaced. So that's already by itself is lowered the cost quite a bit. That's my student actually doing these days. So but things like that we can definitely talk to the institute about open source, what may be possible.
One other quick follow-up on that. So how many, when you said a limited number, is that a limited number per year or a limited max? So if how many Chano systems would Woods Hole be prepared to manufacture effectively on contract? And perhaps equally important, how would you envision technical support and training as part of that? So it's one thing to give somebody something in a box. It's another thing to ensure that they're properly, suitably trained to operate it, and that when it breaks down, they've got a number to call and somebody responsive on the other end of it. So any thoughts about that?
Yeah, this is the model we're trying to build with Northeast Fishery. So the idea is, we build a sensor as based on the request, and then we're gonna test it together with the user within limited, given limited time frame. And then after that, this is not a commercial sensor, so there's no anything. But however, you could build into some sort of maintenance cost so that you can actually we call it service sensor together with the user, and then we're gonna have like a small workshop to train whoever the user are. Then they basically take away what's the major things need to keep the sensor going and the maintenance. So it's not a guarantee in terms of but it's not a warranty commercial service. However, there are like this exactly the kind of the model we try to do with Northeast Fishery, that we do have the budget. When we budget the sensor, there is a small budget that we can try to service the sensor. So in a sense, it's similar to the warranty that in the commercial world, but we just cover people's time and the parts.
So I want to keep the discussion going. I wanted to bring up the option that we may run a little bit over the theoretical 2:30 ending time because we had so much material to cover today. And so we may go 10-15 minutes over if our speakers are willing, and if anybody has to jump out, that's okay too. And I also want to make sure, Sam, I know you had a question, and I want to make sure we get to that.
Chris, is it okay if I ask anybody, or are we sticking?
Oh yeah, I think we can jump around.
Okay, that sounds good. Well, I have one question for one for Aleck and one for Doug. What my question for Aleck was about the EMOLT, and the fishery, kind of observing network, and I was wondering about kind of the time span with which that data might be available, like near real time sort of potential, capacity. Like how long do the fishermen usually soak the traps for? Is there like a typical time period? And so, because the spatial coverage was really impressive for the temperature, you know what I mean? So that seemed cool. And I was just kind of wondering how long, they spend down there subsurface you know, to kind of speak to like the potential, you know what I mean, for monitoring, especially these longer events like Doug talked about. Yeah.
Yeah, that's a great question. As far as I know, the different fishing gears have a different kind of time span. The trawlers, you know, you do, you know, hours, and, you know, then you retrieve it. And once the sensor on board, it wirelessly communicated to the ship computer. And, you know, if it's within the cell phone range, it will just directly, you know, transmit it to cloud. So it's pretty quick. For the trap, for example, they can stay there from days to weeks, okay, and then after that they retrieve it, and the data will be doing the same thing, you know, transmit. So that infrastructure already there. So I mean, imagine a lot of time for the traps. We talk about after a few days, a week or two, then you will have the raw data downloaded. And then for the trawlers, that's probably quicker. So that's my understanding.
Cool. And then for Doug, I guess I was wondering about, you mentioned the 1976 event, you know, like as a corollary, and I was wondering, you know, that one I thought was an upwelling event, like if I remember correctly, that people thought that off New Jersey there was a like a significant upwelling event that happened, but I think there's some, I don't know that anybody's, anyways, but there's obviously, they're pretty close to the Hudson River outflow as well. So there's potential other drivers there, but it was notably very wet this summer, right? And you pointed it out, the cloudiness and the precipitation and whatnot. I guess I was wondering, as you're, you know, obviously it's too soon to sort of assign drivers, but you know, you talked a lot about the low CO2 and stuff like that, but you didn't talk about this sort of freshwater, you know what I mean, kind of lens that might exist in the Gulf of Maine, and if you guys were able to like also co-observe that, you know what I mean, like within this, and if there was any kind of a signal there.
Yeah, there are signals. It's as I said, each part of that is sort of a puzzle, and so the initiation of it is one thing, and that was early April, and that's before the freshet, and that's before the fresh water that we've had this summer. And so the initiation was probably not that influenced by localized freshwater. But then as the summer went on, of course, a lot of the brown water near the coast that people were seeing was runoff. It wasn't this. And so there's a mixture of people that are like concerned about brown water, like in their harbors, and then this bloom. And so those, I think they've been conflated at times, but you know, it's a very small community that's trying to say anything specific. But in terms of this year and why this occurred, I was talking for a while with Dave Townsend about that a month or two ago, and he's just like, don't even try. But I think that we have tracked basically cold water from the Scotian Shelf in February, coming across the Gulf and arriving right about the time that we saw this in Wilkinson Basin, and the ocean color and the cold water filaments line right up. So where there was really cold water, it was greenish brown. And so my speculation is that this came across on the surface, but that's just speculation at this point. And then, yeah, the summer as it progressed, I think we had a mixture. Why it sustained could be tied to the freshwater, for example.
That's really interesting. Thanks. I, as a note on the oxygen side, I have a student working on finalizing his paper. He's so close to submit about oxygen trends and analysis of a bunch of oxygen data in the region. And one of the data sets that he's been analyzing is a really long-term mooring for bottom oxygen around 50 meters, the A1 mooring, right, in the Gulf of Maine. And that has about 20 years of data that's available on the web. And you can look at that and sort of see, relatively speaking, where this year fits in, you know what I mean, to like things. And it's not nearly as low as that 2020 event that killed the lobsters, you know, that people have written papers about and that have been highlighted inside Cape Cod Bay. It's actually pretty high. And I'm a little shocked, actually, because we've been watching it, you know what I mean, because of the event, like knowing it. Anyways, I pulled it up when you were talking too, just to like kind of double check that it still was. And so you can see that it's not anywhere near hypoxic, but no, it wasn't near hypoxic either before the 2020 event, but it was much lower, you know, like 3 milligrams per liter according to the units that are there. So
yeah, and I think what we saw this year is along that like 60-meter isobath where all the Nirkus buoys are, the tide is pretty a strong control there. And so we didn't see the oxygen moving down at our buoy either, or at buoy B. I guess buoy B doesn't have oxygen anymore, but we were keeping track of it where we are. And the other thing is we saw that near shore, this thing sort of came down pretty quick compared to out in the middle. But I do know that MWRA tripped oxygen in late July. They went below anything they've observed in the middle of Mass Bay, which is a little south of A.
yeah,
So
patchy.
it could be you're in the Merrimack plume more. And so it could be that, you know, there is a lot of spatial variability in where these things set up and stayed as well. And we saw similar levels out in the western edge of Wilkinson Basin. We see like 55%, 50%. And so I think it is a little localized to where they were setting up camp, and it was so calm, they really weren't vecting or mixing that much.
So, Doug, just to comment on the MWRA data, it's low. It's low this year. In July survey was kind of the last survey that we have finalized data for, and it's low. 6.2, I think. It's Stellwagen Basin at the deep site, and that's I don't know if it's the lowest we've seen, but it's very low for this time of the year. And it was kind of interesting, the prelim data from the August survey, which is just a couple weeks ago, we expected it to go down, but it was about the same. But that's preliminary. You know, and we've never really had issues with dissolved oxygen. We kind of have a reporting threshold if it's below 6 at Stellwagen Basin.
Oh, interesting.
I'm
That's
sorry.
pretty high. Yeah, sorry, that's interesting. Yeah.
Yeah, it's pretty high. You know, I would argue it's, you know, there's a lot of reasons to argue why it might be lower. But anyway, it is set at 6. And it's something that we've never really had to report on until a couple years ago when we started getting readings below 6.
Yeah.
And last year we had a very interesting sort of what appeared to be a regional event where we got pretty low DOs throughout Mass Bay at the sort of the beginning of the fall, late summer. Which we really haven't thought about too much, but it was, I had never seen that particular pattern before.
Thanks.
So Jake had to run, but he put a question in our little document that crossed my mind too. So I think it's more directed at both, well, it's directed at anybody, but what do you think this event and our ability to understand and track it tell us about the existing observing system and its capacity to respond quickly to unexpected events? And what changes or additions could we make to improve that capability? And so, I mean, that can be for Doug, that can be for David. But what, you know, as we as a group are thinking about developing or improving a regional monitoring system, well, how did things do in the context of this event, and what would help out more in the future?
So I can start, I think, Chris, by, you know, some of the examples I gave in my presentation, you know, I should emphasize that everything we do is required by something or another, whether it's a discharge permit or an MOU with another agency or something like that. So in that sense, it makes it really easy for us to do rapid response stuff for certain things, like Alexandrium or, you know, a plant upset or something like that. You know, I don't know for this Tripos bloom, this is not my area of expertise at all. You know, we didn't do anything extra for it because there was no requirement for it. But Doug is also telling me that the data is really valuable, so that's also great. But I know a criticism of our routine monitoring from our science advisory panel is that it's not temporally intensive enough. And our rejoinder is, we're a wastewater agency. We're not a basic research agency, and these things do cost money that we pass on to our ratepayers. So it's a slightly different, it's easy to talk about things, but a lot more difficult to get things changed just because of that reason, which I do think is valid, by the way. I do want to say that. So, if something totally unexpected comes up, I suspect we're in the same boat as as the rest of you. It's going to be hard to find funding. We're going to have to really come up with really good reasons to get that money to do extra work.
I mean, I think that's one potential answer, especially to somebody like Jake, is money, right? That
It's always money.
Yeah, rapid response funds and their availability is perhaps an issue or something that could be leveraged in the future. All right, I don't know, Doug, did you want to add anything to that?
I don't really think so. I mean, as the others were saying, we were sort of in routine monitoring mode, right? We don't have any rapid response capabilities, or even typically not interest, but that's not something that's in our portfolio. And even what they call ISMN, I'm vaguely familiar with it. I know that we have a project under that, but what you guys with NECAN, for example, and ISMN, it's sort of like every event might be special to somebody like me. I want to know where my carbon's going. But I think HABs, for example, that's a very specific customer that is paid for and regulated. And this one didn't fit into that. And so I think event by event would be part of the story. But I think for fisheries, this one could be significant, because you don't know what this means for next year's zooplankton crop, for example. And so if this starts happening again, this may move into an ecological event that we need to keep track of. But, I think every event could be different, So that's all I can say very quickly. The money part is less important to me than who does it and what, how big does a group need to be to do it correctly? And I don't have answers to those things.
That's fair enough. We were kind of making it up as we go along, right? Let's see. I know, okay, in the chat, and I think this relates back to the discussion we had earlier with Aleck about commercialization, but Mike Brosnahan, who I don't think he's on anymore, I don't see his name list, said, he just mentioned that there's a hybrid path that's been explored through marketing of imaging flow cytobots with McLean Research Labs here in Falmouth. So I think sounds like there's some sort of partnership where a company handles the marketing, but perhaps somebody else does the production and support. And I saw Adam Pimenta has his hand up. Adam, you want to throw something in the mix?
Yeah, this is a question for Doug Vandemark related to that bloom. So I've seen a lot about high temperatures in the North Atlantic, and I was just curious if the Tripos bloom and the low PCO2 conditions were related to higher than normal temperatures in your observations.
I think the answer is yes, because they're correlated. Related,
yeah.
But they're correlated because it's just warmer. So even the winter water on the Scotian shelf was much warmer than usual winters. And so it's certainly correlated, and I wouldn't make any speculation past that right now.
Okay, thank you.
I think David had his hand up, didn't he?
Yeah, I just wanted to briefly mention that there is a science advisory panel to our current discharge permit that's being disbanded. They've issued a draft permit, but EPA has been talking about a regional sort of, not necessarily Gulf of Maine wide, but Mass Bay wide sort of, I don't know, consortium, I guess, to sort of handle, because they felt like that MWRA was kind of carrying a lot of load in Mass Bay. And they wanted to, I don't know, it seems like they want to spread it out a bit, but it might be something that you folks might want to sort of keep track of. It's very much in its infancy at this stage. But they were thinking about running it through the Mass Bays estuary program. But, this kind of fits into this, coordinating, different data sources and sample collectors and things like that. I don't know anything more about it than what I just said, but it seems like their intent is to try and set something like this up. And I know Jake and I talk occasionally every few months, and I did mention it to him last time I talked to him.
All right, so we've kept everybody 10 minutes past when we said we would. I wanted to give steering committee members one final crack at putting a point out there if they want to, before we thank our speakers and set everybody free.
Sorry, I have my hand raised, Chris.
Oh, sorry, Aleck, I missed that. Yeah, please do.
I just want to mention, I think, as I mentioned, the fishery-based observation can be used for rapid response. And I believe people are doing it for hypoxia. I think the Tracy Pugh and the Massachusetts Division of Marine Science, they are actually have this, Cape Cod Bay study fleet, and they monitor the oxygen using, low-cost sensor, and that give pretty good, spatial and temporal coverage, even though the data are not real time, but it's quick enough to respond because a lot of the sensors are being retrieved. I mean, the data being retrieved every few days to weeks, so that can give somewhat real time or near real time reaction. Though, what would be the reaction? It's not clear to me. What are the action? People look at the data, what they're gonna do with it. So that's it's not too clear to me. By talking to Tracy. So, I just wanna. That's a thought.
That's a policy question that I don't even want to get close to, Aleck. That seems like a bag of worms, but no, I mean, that's very fair. But I think the capacity for sort of citizen science or science of opportunity using something like a fishing fleet is a really compelling case that NECAN can perhaps look at as we develop the plan. Okay, we've kept everybody for quite a long time. Sorry, Cam has his hand up.
Sorry, I missed some of that discussion. I had to step out to go collect my child. But just in that last bit, when thinking about a rapid response, I actually think that UNH's role in this latest event actually really showcases the value of routine monitoring as well, and being able to respond to an event like this. And in the sense that the response was almost more of a focus of our attention on the changes being made. And because we were doing the routine monitoring, we were actually able to see all the changes that were happening in the ecosystem. And we even made this point when we were talking to NCOS about possibly getting some more funding to do some more survey work. We tried to make the argument that no, rather than spreading out our efforts, we could support the routine monitoring a little bit more to do a little bit more surveys and adding a few instruments, because it's not a data point of one. It's like we're actually able to take what we've learned from years of doing monitoring at this station, and then and just adding on to that bit more with a bit more resolution. All right, that's all I really had to say about that.
Right, I think that's a great place to leave it. Austin, did you have a final wrap up that you wanted to give?
Yeah, I just want to say thanks again for everyone who stuck out for these last 15 minutes, and to all of our presenters for those wonderful presentations. This was the final webinar in the 2023 webinar series. So keep an eye on your email for whenever the 2024 NECAN webinar series starts up. Thanks again, everyone, and have a great rest of your day.